Hello, everyone. Good day to you all. We are going to get started in just a moment. We will take a beat to let the Zoom let everyone in. As you are joining, feel free to say hello in the chat. Let us know where you're joining from. It's a rainy spring day here in Michigan. Okay, I'm going to go ahead and get started. Welcome, everyone. This is Christy Sullivan. I'm the Secretary of the American Society for Cellular and Computational Toxicology. This is our second April webinar for 2024 and the third in our series of webinars highlighting the work of award winners from both the ASCCT and the ESTIV Society. I want to encourage you to visit our websites to learn a little bit more about both of our societies and support our activities, namely this webinar series, our annual meetings, and the awards and support that we provide to early career scientists and students from all over the world who are working in the NAMS fields. You can do that by becoming a member of either of our societies. The webinar is recorded and the recording will be posted on the ASCCT website webinar archive a few days from now. In the webinar platform, I'm sure you're familiar with by now, but you can ask questions using the Q&A feature at the bottom of your screen. Please don't put questions in the chat. It makes it easier to keep track of them all if they're in the Q&A. There will be a Q&A after each speaker is finished, so while they're talking, feel free to get your questions in. We have also enabled closed captions, and you can turn them on or off in the toolbar at the bottom of your screen according to your preference. Before we get started, I want to give you a couple of brief reminders. So first of all, the ESTIV Congress, which is happening this year in June in Prague. Everyone's very excited for it. Today is the last day for early bird registration, so get on over there and get that taken care of if you haven't already. The ASCCT 13th annual meeting will be in the fall in Research Triangle Park, North Carolina. And we are still accepting abstracts until June 30th or even until September 1st for posters only. So feel free to visit the website and check that out. And then finally, tomorrow is the deadline to apply for an ASCCT-sponsored travel award to the 2024 Summer Immersion on Innovative Approaches in Science. That's a Physicians Committee-supported program that's going to be in Washington, D.C. in late May of this year. So please visit our website to find out more about that. We have the next webinar scheduled for May 14th, but we don't have the registration up yet. That should be getting up fairly soon. But in the meantime, we wanted to let you know that there are quite a few plans for upcoming webinars in 2024, including on all the topics you see here and more. So please consider becoming a member of either ASCCT or ESTIV to make sure you find out about these upcoming webinars. And I will go ahead and introduce our speakers for today. Our first speaker is Damaris Cristine Pinto. She is a biotechnologist and a human environmental safety assessor at Grupo Boticário, a leading Brazilian cosmetics company. She was winner of the first ASCCT International Travel Award in 2023 for her presentation on assessing sunscreen product toxicity on coral fragments, a new approach methodology for enhancing reliability and addressing environmental concerns. I'm going to introduce our other two speakers and then I'll turn the floor over to Damaris. Jerry Achar is a PhD candidate at the Institute for Resources, Environment, and Sustainability at the University of British Columbia. in Vancouver. His research spans from chemical toxicity prediction using QSAR models to analysis of uncertainty associated with the models and model predictions. His latest research publication proposes a problem formulation framework that characterizes components relevant to the application of in silico toxicology methods for chemical toxicity prediction. And Jerry won the Suzanne Fitzpatrick Student Travel Award from ASCCT in 2023. And finally, our third speaker is Kirsten Veltman, who is a third-year PhD candidate at the National Institute of Public Health and the Environment and the Leiden Academic Center for Drug Research in the Netherlands. In her PhD project, she focuses on next-generation hazard assessment of non-genotoxic carcinogens, including oxidative stress, using both in vitro and silico approaches. And Kirsten won the ESTIV Best Poster Presentation at the Eurotox 2023 Congress in Ljubljana, Slovenia. Okay, I will go ahead and give the floor to you, Damaris.
Thank you, Christy, for the introduction. I hope you are all seeing my screen right now.
Everything looks good.
Perfect. So first, thank you for the opportunity for being here. For me, it's a great pleasure to be here representing Grupo Boticário and the entire team involved in this work. And today I'm going to talk about assessing sunscreen product toxicity on coral fragments, a new approach methodology for enhanced reliability and addressing environmental concerns. So first, just to give you a little background. So Grupo Boticário is one of the biggest beauty franchise in the world and one of the 15 biggest beauty companies. And we are proud to say that we are the third most sustainable according to the Dow Jones Sustainability Index, released on 2023. We have more than 15 brands, including the most loved brand by the Brazilians, O Boticário. And because taking care of the environment, it's very linked to our purpose, which is to create opportunity for beauty to transform each one's life and thus transform the world around us. We launched in 2021 the movement Beauty for the Future, which is a ESG, so environmental, social, and governance commitments that will be delivered by 2030. And among these goals, the protection of the aquatic environment is one of the goals that we have. So by 2030, we will guarantee a lower impact on the aquatic environment for 100% of our rinse-off products. So because we believe in our responsibility as change makers, the protection of the environment, the aquatic environment is very important. And to keep health in aquatic environment, the coral safety is an important issue, an important topic, because coral reefs plays an important role for keeping the marine ecosystem and biodiversity. But year after year, we can see in the news and the newspaper news about the coral bleaching. The coral bleaching is a complex issue that can be influenced, and it can be influenced by different factors like global warming, pollution, and other factors. The global warming is the main cause because the thermal stress, like the water heating, leads to the breakdown of the symbiotic relationship between coral and their photosynthetic algae. But Other local stressors can influence in the coral bleaching process, like intensive agriculture, fishing, poorly treated wastewater, and chemical pollution. So regarding chemical pollution, sunscreens emerge as a possible pollutant because sunscreen can enter on coastal areas indirectly through the water from treatment plants, or directly because people are using sunscreen to go to the beach, or because this sunscreen can be directly in contact with the marine environment. And some studies have indicated that some organic UV filters can have some toxic effects on corals. These studies are very controversial. The scientific community remains uncertain about the extent of the effects that was seen in these studies on the nature. But these recent studies have led to the ban of some controversial ingredients that are present in this, some controversial UV filters. So the bans in Hawaii and Palau, Virgin Island, of oxybenzone and oxyocyanate. And the ban of these ingredients leads to a consumer awareness because people want to use a product that is environmentally good. And this consumer awareness create a new marketing demand for reef-safe products. But there is no guidelines on what is a reef-safe product. And it's not possible to say that only removing the controversial ingredients that this product will be a truly reef-safe product. So this creates an ambiguity between industry and the consumers, and we are seeing some lawsuits. Some lawsuits of companies that claim that they claim themselves as reef safe, that their products are reef safe, based on removing the controversial ingredients. So to address this challenge, what we can do and what we propose here. So we propose a new approach methodology for assessing sunscreen product toxicity on fragments of Ceratophora hydrix, on a coral fragment. So by using this new approach methodology, we can assess the entire product formula. So we have a more holistic perspective and a more reliable assessment of the toxicity of the product. And we can establish a clear criteria for a clear criteria for defining what is a reef-safe product. So this can support regulatory agencies and companies. So here to introduce you about how this methodology works. So the study organism is Ceratophora hydrix. So these fragments are not taken from the nature, so they came from a controlled environment. This species, Ceratophora, it's a small polyp stony species, so it's known to be a reef-building coral, and it has a wide geographic distribution. For this study, we used 7 sunscreens with different sun protection factors. And to guarantee that the product were not toxic for these coral fragments, the endpoints that were observed was fragment retraction and the coral fragment bleaching. And to guarantee that these test organisms were sensitive enough for the analysis, we performed a sensitivity assessment using copper sulfate, which is known to be toxic for corals. So here is explaining about the methodology. So we have the samples of the sunscreen, so the entire product, and we diluted these samples with seven different concentrations. The lowest concentration is 3.2, and the highest concentration is 100 milligram per liter. So it's important to highlight here that this highest concentration that we used for this new approach methodology is according to what is adopted by the OECD ecotoxicological protocols for defining that a substance is not toxic for the aquatic environment. So after diluting the samples were went through a water accommodation fraction process. This process is indicated for samples with a low solubility to guarantee that the samples were well homogenized. And after the water accommodation fraction, the samples were put in contact with the coral fragments. So all the entire test is done in replicates. So the samples are put in contact in triplicates for 48 and 96 hours. Important to highlight here too that we have a negative a control too, so organisms that were put in contact with a medium with no samples, like sugar as well. So after the samples stayed in contact with after the coral fragments staying in contact with these samples for 48 and 96 hours, we observed the endpoints, the retraction and the bleaching. And after that, photos were taken with these photos, we perform a R-gene-B analysis. So this R-gene-B analysis is for the quantification of the color variation with the beginning of the study after 48 and 96 hours. So this color variation can indicate for us how badly or the bleaching process was. And this assessment was performed using Photoshops. And after that, so after the endpoints observations and the R-gene-B analysis, we performed a statistical assessment to calculate the effect concentration, the lowest effect concentration, and the no effect concentration using a statistics software. So this methodology was developed in a partnership with an environmental lab here in Brazil. And this methodology was based on a already published work that was done by Canaves and colleagues. And the difference between this new approach that I'm showing for you today and the work that is already published is the incorporation of this R-gene-B assessment, the color variation, the quantification of the color variation. So an increase in the range of the red, green, and blue pattern can indicate for us a greater bleaching, an increase in the range of the RGB value can indicate a greater bleaching. So this protocol does not fall under the definition of the animal testing according to the European, American, and the Brazilian regulation. And to guarantee that this, to make sure that the results was considered valid, so two criteria must be met. So the first criteria is that we cannot see in the at the coral fragment, we cannot see no polyp retraction. So here, let me point to you. So here in this picture B is what is how a polyp retraction looks like. So in the fragments with no samples, with no sunscreen samples, we cannot see polyp retraction. And for the sensitivity assessment using copper sulfate, so this image here, the results that we obtained in the sensitivity assessment must be according to the must be according to the chart control. So the control chart that we had that was already established for this assessment. So for these results that I'm going to present you, so these two validation criteria were met. So here in this table, we have the results for after 48 hours of this sunscreen samples in contact with the coral fragments. So here, based on visual observations, were not based on the visual observations, no retraction and bleaching was observed even at the highest concentration. So for all the samples. But for the RGB assessment, so the color change variation, significant color change was observed for one sample here. So at the highest concentration, so the lowest effect concentration was 100 milligram per liter, and the no effect concentration 56 milligram per liter. So after 48 hours for 48 hours for one of the samples. So all the samples showed no adverse effects after 48 hours. So even at the highest tested concentration, so based on visual observations. And one sample had an increase of RGB variation at 48 hours when compared with the control. So the color, something that is important to highlight here, and that's why it's important to measure this color change, is that visually this color change was not possible, like for as a person to distinguish, but based on the RGB variation, we could identify the start of this bleaching process. So after 96 hours for two samples, we had effects. So for 5 samples, we had no effect after for the highest tested concentration. So, and for two samples, effects was observed after 96 hours based on visual observations and color variation. So the highest tested concentration was 100 milligram per liter according to the OECD guidelines. And the products that were not, that had no observed effects and significant color and a color variation that was significant, like they were claimed and they are claimed as reef-safe, so the products with no effect for the visual observation and for the RGB assessment. So only those products can be claimed as reef-safe. So 5 samples had no adverse effects at 96 hours, while 2 samples were toxic. So the effect concentration for the sunscreen, sun protection factor 7.70 was 23.8, and the sunscreen sun protection 30 was 7.55. So the RGB data analysis confirmed that the fragments in contact with the samples that were toxic displayed the most substation color variation through the study. So these visual observations can be seen when we see those visuals. It is when we see bleaching and retraction, we can see a more color variation in the RGB assessment. And the samples with toxic effects had no UV filters or preservative at or below the concentrations of non-toxic formulations. So that was a big surprise for us, because we can think, oh, so probably the samples with the highest amount of UV filters or preservatives probably are the most toxic samples. But this is not what we observed through this study. So here is just to highlight all the UV filters and preservatives that we used that were present in those samples. So it's not possible to say that samples that were toxic had more had a more concentration of UV filters or preservatives. So identifying the root causes of the toxicity of the samples remains a challenge. Right now, we are performing ongoing studies with our analytical lab to explore this further. And this just highlights why it's not possible to say that a sample or a product is reef-safe based only on removing controversial ingredients. Because a formula, it's a complex mixture, so evaluating the entire product, it's very important. So simply removing the controversial ingredients is not sufficient to guarantee no toxic effects on coral. So testing the final product, as we are proposing here in this new approach methodology, is crucial. So by adopting this new approach methodology, this can be a robust and reproducible assessment for labeling a product as reef-safe, not only removing the controversial ingredients, and by using this assessment, this scientific-based assessment, we can meet the consumer's demands for reliable and reef-safe sunscreen, and we can support companies and regulatory agency for a scientific-based comproval, substantiation of the claims of the product, the attributes of the product. So thank you for the opportunity for being here. I want to thank you first, ASCCT, for the opportunity to be here today, and the Grupo Boticário R&D and Safety Department, and the Safety and Environmental Insurance Department.
Thank you, Damaris, for that really clear presentation. I was fascinated by the results and have a lot of questions myself, but we'll go to the questions here from the audience first. So one question is about the coral species. How representative is it of other species of coral? Do you know?
Laura, first, thank you for your question. For choosing this species, we choose a species that is a species that is reef building and a species that has a wide geographic distribution. So for have a more realistic assessment. When we are talking about toxicology assessments, like for performing, we need to choose a study organism, and this result we understand that this result will be similar to other similar species, or this is performed the same for other kind of toxicology assessments.
Okay, thank you. And Laura's also wondering about chronic exposure. Has anyone looked into that? So exposing the coral for a number of weeks instead of hours.
This is more like because the coral because the symbiotic algae that lives with the coral, they have like a very fast metabolism, and for performing acute and chronic assessment for algae, usually the time frame is 96 hours. So the same time frame that we are using here.
Okay, very interesting. We'll take one last question from Stephen. They're asking about the experimental unit. So it seems like there's a batch of sunscreen. How many batches are tested, or is it just one?
It's milligram per liter, and this study is done in triplicate. So for each concentration, we perform in three batches in triplicates.
Okay, and I also think maybe their question is getting at, sometimes you do toxicology testing on multiple batches ongoing because you're afraid there's some kind of contamination, but that's not quite what you're doing here. Here you're looking for the ingredient toxicity assessment, right?
Yes, perfect.
Okay. Well, thank you again for your presentation. We are going to move on to Jerry's presentation. And if more questions come in, Damaris can answer them in writing. So please feel free to add your questions. Hello, Jerry. I see your
Thank you, Kirsten. Can you see me?
outline?
That's the outline. Let me see.
Yeah, I see your full screen. Looks good.
Thank you. Thank you. All right, everyone. Thank you, Christy, for inviting me to present today and the organization too. I'm gonna present today part of my research. This is a big project, so I wasn't able to put everything out together to present today, but it's part of what I presented last year. It's expanded a little bit thanks to the feedback that I got from the conference. So the study is analyzing implicit and explicit uncertainty in T-cell prediction of toxicity of chemicals. And I'm going to explain in a little while what implicit and explicit uncertainty mean in this context. So just a little bit background, maybe probably what you already know, there's a growing appreciation of the potential to apply in silico toxicology methods, including QSAR, in the prediction of toxicity of chemical compound. And one of the emphasized needs is to apply it for complex endpoints, including neurotoxicity or developmental endpoints or such like endpoints, because the animal model predicting or testing such complex endpoints sometimes are branded not being as reliable. And so kind of like having something like QSA to supplement or to fill data gaps is one of the, present needs that are recognized. But the problem, or one of the challenges that I experience, is the uncertainty that are attributed to that we kind of see in low regulatory uptake or acceptance of the prediction. And this uncertainty usually spans from, the data that is used in developing the models, justifying similarity between molecular descriptors that are used in developing or in the prediction processes, or just like the relevance of the model. Sometimes it also goes up to like the algorithm or the model structure and, the scalability of such algorithms to perform the intended prediction. And interpretation also counts with that it also includes mechanistic causality or interpretation of the prediction. And so in this research, we kind of aim to identify uncertainty. So our curiosity arose here from when we started like reading publications on some of these complex endpoints. And we chose neurotoxicity because presently there's no sufficient data on animal testing. There's a great call to apply in silico toxicology methods like I mentioned to fill the data gaps. And it's a complex endpoint, so it means that only a few QSA techniques have been developed to this end. And so our hypothesis was like, if we don't have enough animal data on this, and the application of QSA is just taking off, then there's a high possibility that we still have uncertainties that are associated with the models and the model prediction. And so what we started doing here, we were like, let's analyze publications that have predicted neurotoxicity of chemicals using QSA and, identify some of the uncertainties that they talk about explicitly, directly, or implicitly, those that are hidden within the text or statements that authors or publishers, usually express in such publications to see some of the uncertainties that they talk about. And again, as I mentioned, our study data material here is like peer-reviewed publications, looking at, specifically publications that apply QSAs in the prediction of neurotoxicity of chemical compounds. And again, when reading the papers, we hypothesize that there's a high possibility based off our reading that the uncertainties that are communicated explicitly, for example, someone will say that we are presently uncertain about this, or there's a data gap in this part here. But there are also those that are implicitly communicated, and this is based off linguistic application or use in expressing or communicating study findings. So like it's demonstrated or illustrated here in the figure, we were like, there's a lot of hidden uncertainty. Some are expressed implicitly, some are not included. So that's like a unknown. We didn't get there because we may not know what was the uncertainty that was present yet was not included, or some that, they just failed to communicate. So our interest was in the first bullet, like to kind of like assess or investigate uncertainties that are there in the text, but are just implicitly communicated. The process that we undertook to analyze or identify the uncertainty started from, we identified the papers, peer-reviewed papers, publication, QSA prediction of chemical toxicity. Then we did in a group of 4, I did line by line text coding and analyzing text to identify uncertainty indicators. Those are the expressions or descriptors that indicate uncertainty. Could be explicit, like I'm about to explain, and or implicit. So implicit here simply means that sort of phrases that indirectly express uncertainty. So it could be the use of may, maybe, likely because, possibly, attributed to, among others. And explicit are those that you can easily read the text and directly know that this is uncertainty being communicated here. So that include, just directly admitting that, we are uncertain about this, or there's presently no data on this, but just direct depiction of uncertainty in a text. So we kind of analyze both and categorize the indicators. After that, after recording the final list, we analyze the context in which the uncertainty indicators are used. So this is based off how the indicator relate to the main or the central information communicated in a text, and how the indicator influence or impact that central information communicated in a text. So to do that, to understand the context in which it is used again, we had to read the whole paper, narrow it down to the whole paragraph, and then analyze or just determine how the indicator influences, an indicator influences the central information communicated in a text. So after then identifying the uncertainty using the uncertainty indicator, we move to the next step to categorize the uncertainty based off the different categories that we develop. So this is just a demonstration how the process, we undertook the process. I'm going to use the user here. So the first column here shows how we went about identifying implicit uncertainty. So look at this sentence. So it demonstrates that a small molecule is more likely to cause adverse effects to nervous system. So within this sentence, the uncertainty indicator is more likely. So this indicator here, it indicates uncertainty, but it doesn't directly indicate uncertainty in the sense that these people don't directly admit that there's uncertainty, but based off linguistic interpretation of the use of more likely, we interpreted it that it's implicitly communicating uncertainty. And so the process started by identifying the indicator itself. So in this case, more likely is the indicator. And then the next step involved identifying the central piece of information communicated in the text. So, and again, like I mentioned, you have to read the whole paper, narrow it down to the whole to the paragraph where the uncertainty indicator is to understand the information. So in this case, in this example, the central piece of information communicated is like a smaller molecule causes more adverse effect to the nervous system. And if you bring in the indicator, then it means that the indicator modifies the central piece of information communicated here, which is like it's not always guaranteed that the smaller molecule will cause adverse effect than a larger molecule. And this is based off the interpretation of what they communicated in the old text. So in general, the interpretation of this is the uncertainty is there about potency and mechanistic plausibility of the chemical, that it's not certain whether it has adverse effect. Again, the mechanistic interpretation of that is not certain. So that's the uncertainty communicated there. So if we look to the right, where we have the explicit uncertainty, so here indicators that directly communicate uncertainty. In this case, the sentence is talking about, unfortunately, there's not enough experimental data to corroborate these findings. So the use of there's not enough experimental data, it's directly communicating uncertainty. So we took this as an explicit uncertainty indicator. And as with the first column, we had to understand the central piece of information communicated, which is like a group of chemical found to induce neurotoxicity in the test animal, which was mouse. And then if you bring in the indicator, then it impacts the message communicated here by directly admitting that supporting evidence of the observed neurotoxic effects of chemical is insufficient. So our interpretation of this was this uncertainty is related to the elicited potency, the evidence that supports the elicited potency. So after having analyzed the text and identified the uncertainty, we came to the next step where we had to categorize the uncertainty. So this is a framework that we developed in our previous research. So it outlines different modeling phases, the one that are in the oval shapes. And then the modeling phases, the model creation, model characterization, model application are linked to modeling components, which is data, mechanism, performance, kinetics, relevance, applicability, and the rest. So these are the components that are kind of linked to different modeling phases. And then just down there, we have sources of uncertainty. So the sources of uncertainty are the ones that are in the dotted boxes. So under each component, there are different uncertainty sources that we can characterize within them. So we're not going to go into defining each uncertainty source here, but basically what this means is that different uncertainty source has different coverage. For example, uncertainty source here simply means areas where uncertainty resides. Like for example, if you talk about data quantity here, it simply means that uncertainty related to the amount of data used in modeling, that could be in model development and validation. Or when you talk about something like model structure, simply means that the algorithm or the mathematical formula used in a model, something like that. So we kind of like mapped our interpretation of the uncertainty identified from the text against the uncertainty sources. So we kind of categorized the uncertainty identified from the text we map them onto the uncertainty sources here. So the uncertainty presented in this study is represented as uncertainty sources. So for example, in this case, if we have uncertainty in the first column related to potency, then we categorize them as uncertainty in activity or potency of that chemical more generally. And part of the findings from our analysis is that different uncertainty, so this is a relative frequency of different uncertainty sources as identified from the text and mapped against the framework that I've just shown you. Mechanistic plausibility ranks the highest, followed by a number of highly ranking uncertainty sources. So that's descriptor concordance, we have model relevance. So these are the areas that are highly talked about in the analyzed text. So we kind of like looked at the number of occurrence of such uncertainty sources and then calculated the relative frequency. And this shows like in ascending order from data validity up to mechanistic plausibility, the relative frequency, how they occur. And based off this, we transform the relative frequency to understand the level of uncertainty themselves. And from here, we use a 3-point uncertainty ranking scale to, in attempt to kind of like just transform them to understand them better in terms of what are the level of uncertainty, how can we characterize them? And in doing this, we would like to note that there's no definitive way of doing this, but in the uncertainty characterization literature, a 3-point ranking scale for uncertainty is not uncommon, but there's no like kind of definitive scale on how to do this. So we tentatively did this just to help us interpret the data that you have from the previous slide better. And you can see that the areas with high uncertainty levels are the 5 areas as we saw from the previous slide, and then you have moderate areas, and then you have low uncertainty. So the takeaway from this is that we have, best of the categories, we have different levels or concern about uncertainty. Some are high, some are moderate as for now, and some low. And this is based off our interpretation of how the authors of the studies communicate uncertainty. And then we went to individual analysis of uncertainty, implicit versus explicit. Implicit uncertainty here means those that are kind of hidden within the statement, those that are indirectly communicated in the text, and explicit here means those that kind of directly communicated in the text. And from the graph, we can see that consistently, implicit uncertainty are higher. They have higher relative frequency compared to explicit. Apart from a few, like in this case, we have data quantity, which has higher explicit compared to implicit. But generally, the conclusion that you can draw from here that uncertainty is highly communicated implicitly. So we have a lot of hidden uncertainty that are not transparently communicated, at least with regards to the analyzed text or the studies that we analyze on QSAR prediction of the neurotoxicity of chemicals. And so this kind of like generally communicate that we only have a few uncertainty explicitly communicated. Something that is very interesting that in all the studies, we did analyze 30 studies, by the way, sorry, I didn't mention that. We did analyze 30 studies, and all the studies, none of them had a clear section or, the set aside section to kind of analyze uncertainty. All of them just had running text just to report study findings, but none had any of qualitative or quantitative reporting of uncertainty. So this is very interesting that though we have uncertainty, only a few of them are explicitly communicated, and none of them are kind of like communicated through a section in a study to quantitatively or qualitatively. And basic conclusion, which is also a summary of the study that we have here, is that presently, from the analyzed text, uncertainty related to mechanistic plausibility, descriptor concordance, relevance, model performance, activity or potency of chemicals are among the most frequent. They are among the most occurring uncertainty sources in the QSAR prediction of neurotoxicity of chemicals. And this can be understood, especially for mechanistic plausibility, because there's in the literature, it's highly admitted that there's a large data gap, especially in understanding the complex morphology of, test animals during predictions or interpreting mechanistic causality of chemicals when applying models. Model relevance is a great issue, how model can be applied outside of the area of prediction. Model performance, again, is also an issue. Presently, to my knowledge, there's no many like QSAR models that have been developed to this end. And so something like, a model being able to have high sensitivity or predictivity in that sense is still very low. External validation is something that is highly talked about in this area. And so it makes sense to have high uncertainty with regards to model performance. Data balance, data accuracy, chemical similarity, among others, had low frequencies. This is surprising though, one, because animal data is, the uncertainty that are reported in regards to animal data that are used to develop this model. So when you have something like data accuracy or chemical data balance not being talked about, it was a little bit surprising for us, or the data validity. One example is, the databases that we also analyzed or just like perused through to see whether they're reporting the test methods used, whether they comply with the OECD test guidelines or GOP. They were never reported. So data validity in this case, whether data is obtained from standard test guideline or not, those were not reported in analyzed studies. So that was surprising for us. So in general, we could say that uncertainty is highly implicitly communicated compared to the explicit uncertainties. And using our indicators, we were able to categorize the uncertainties, analyze and identify from the text. And we hope that the study findings could be, beginning of finding ways to kind of, finding ways to standardize an analysis of uncertainties, especially implicit uncertainty communicated in scientific studies, or verbally expressed uncertainties, and probably define or design ways to kind of address the uncertainties communicated by scientists or verbally expressed in scientific studies applying in silico toxicology methods. And that's it.
Thank you, Jerry. I think you've done a lot of really interesting work trying to characterize the literature in terms of uncertainty. I don't see any questions yet, but we'll wait a moment to see if anyone wants to add a question in. And I'll ask a question. When you were looking at, did you look at papers that specifically talked about or presented a QSAR model, or were you mostly looking at papers that were gathering data to make the model?
So we look, thank you for the question. So we look at papers that, developing the models and applying the models in prediction exercises. Yeah. Right. So, but basically we were kind of like our scope was within QSARs only. Yeah. But it was both development and application of the models.
Okay. Yeah, that's really interesting. Thank you. I also wonder what I was thinking when you were talking about the implicit descriptions, like more likely. I think sometimes our training as scientists encourage us to say something like that rather than something more definitive. Did you think about that at all?
Yeah, we did. We had during our coding process, we kind of had like, it was that, and also, language, the cultural differences in using of language, because we were collaborating with folks from the UK. And one of the question brought up was like, they use, for example, maybe as a politeness, not because of the uncertainty. And so we had to kind of filter those out. But in terms of like, just in terms of how we use the indicators in science, it was so difficult to kind of remove those off, because our interpretation, we could use those when you're not sure, but we could not just remove them based off what we couldn't just think of how the author intended to use it. We were based off linguistic interpretation of the indicator or the expression.
Okay. We have one question from Anna. They're wondering how many papers altogether did you go through?
We did 30 papers, and we couldn't find more than that based off the selection criteria that we had.
Okay, great.
There are not many papers published for QSAR prediction.
Yeah, okay, cool. Well, certainly this is a very compelling topic, specifically for QSARs and also for lots of other types of NAMs, and a social topic, right? About how comfortable people are with uncertainty and how we talk about it. So thank you, Jerry, for your presentation. Thank you all for listening. And we have one more presenter now. We'll turn over the floor to Kirsten Veltman.
Yes, I hope you can all see the screen now.
Yes, looks good.
Okay. So I would like to start by thanking you for allowing me to present my work here today. And what I will present is my work on the application of WGCNA as mode of action screening method for suspected non-genotoxic carcinogen, which is a project I've been working on as part of the Risk Hunter Consortium. So to first put this project in a broader context, I would like to give some small background about my PhD project. So the aim of my PhD is to aid the transition to a mechanism-based human relevant safety assessment of suspected non-genotoxic carcinogen using new approach methodologies. And as you might know, there are many modes of action through which non-genotoxic carcinogens can have their effect. So I specifically focus on oxidative stress leading to regenerative proliferation. I would like to give a small background on the AOP, adverse outcome pathway concept. So AOPs are these sequence of events, or we call them key events, that go from the first interaction of a chemical with the body to the ultimate adverse outcome. And what I want you to know about AOPs is that they are chemical agnostic, which means they are not stressor specific. So as soon as the MIE is activated by a chemical, the body will not distinguish between chemical A or B. It's just about how much this key event is activated or perturbed. So the second thing I want you to know about AOPs is that they can integrate multiple pathways. And this is also now becoming more popular to develop AOP networks. Basically, it means that there are multiple MIEs or a different series of events that can lead to the same adverse outcome. And the last thing I want you to remember about AOPs is that they are a very useful tool in aligning assays for non-animal test batteries. So you can identify NAMs for each block, for each key event, and then link them through the AOP. So I've also worked on the developing of an AOP for oxidative stress leading to cancer. So an important side note here is, despite everything going through this central node oxidative stress, because that's what my main focus is, I'm not saying that there's not another way through which a chemical can induce some of these key events and lead to cancer, but that was just not the goal here. So you can see that there are multiple of these MIEs all going through oxidative stress and then via multiple sequence of key events leading to cancer. And this is really the basis for the work that I am doing within my PhD. So as I said, we want to develop a mechanism-based human relevance safety assessment method or test battery for non-genotoxic carcinogens. And because there are so many modes of actions, we feel that it's useful to start this test battery with a mode of action screening method that can give alerts or directions for further testing. And we think that weighted gene co-expression network analysis, or WGCNA, could be a potential candidate for this mode of action screening method. And again, we are here focusing on a case study of chemical induction of oxidative stress leading to regenerative proliferation. So what is this weighted gene co-expression network analysis? It is a method to analyze transcriptomics data, but instead of studying single genes, you study modules. And these modules are groups of genes with a similar expression pattern across a large number of samples. Because they have this similar expression pattern over a large number of samples, these genes are thought to be either co-regulated, functionally related, or members of the same pathways. So here you can see a schematic of how this works. So all the lines indicate that the genes are somehow connected. And then based on how connected they are, you can identify the modules. And these modules all have what we call a driver gene. And this is a gene that largely influences the overall expression of the module, and it often influences the gene expression of the other genes within the module. And because you have these clusters of genes, this is very suited to annotate for pathways, enrichment terms, and transcription factors, which is very useful in the interpretation of the data. So if you generate these modules for all the genes that you have studied, you get what we call a dendrogram, in which every dot represents a module, and the size of the dots indicates how perturbed the module is, and the color represents whether it's upregulated or downregulated. So for my research, I am using an existing WGCNA map, which is based on the primary human hepatocytes TG-GATES data. And this map has been generated by colleagues of mine in Leiden. So my research is really about applying this method, this WGCNA for hazard assessment. So I did an experiment with primary human hepatocytes, which were exposed to various chemicals for 24 hours, after which the samples were sequenced using tempo sequencing, and then were projected onto the existing WGCNA map. But what I want to focus on for now is the part where I mapped the modules to the AOP and on data interpretation. So first off, the identification of modules of interest. So I just show here a very simplified version of the AOP I showed before, but I actually did this for all the key events that are in the larger AOP. So you start with this series of key events. And for these key events, you can think of enrichment terms and signaling pathways and even transcription factors, which are associated with these key events. And then the next step is to match these enrichment terms, signaling pathways, and transcription factors with the ones that are in the module annotation. And then you obtain modules of interest. But of course, you also need to verify whether these modules really represent the key event you think they are presenting. So I also did a bit of a verification of these modules. So I used some prototypical chemicals for key event perturbations. And of course, I also took along a chemical that should not perturb the key events. And then I checked whether there is an increase in the module response. So the response of the module is called the eigengene score. So if there is an increase in this module response with an increase in concentration. And I also checked whether there is an increase in the module score with time of exposure. And the last thing I checked is whether the modules were significantly perturbed by the chemicals. And based on the TG-GATES data set, we set the threshold for significant perturbation of 2 or -2. And this, of course, reduces the number of modules you link to your AOP. But you have a bit more certainty that the modules really represent the key event you link them to. And the next step is to develop a logistic regression model. So a logistic regression model can predict probabilities of a binary outcome. So in this case, the input is the WGCNA module scores, and the output is then the probability of oxidative stress induction, yes or no. And the reason why we are interested in developing a logistic regression model is that you can then compare predicted probabilities between different modes of action. So then on to some results. So first of all, a big plus for the WGCNA analysis method is that it groups the genes into modules. So here you see one of these modules. This is a module for oxidative stress. And now instead of having to study all of these genes individually, I can just study the effect on this module as a whole. And this greatly reduces the number of entities for your output, but it also simplifies the interpretation. So over here you can see for different concentrations of one compound, the number of differentially expressed genes, which is the black bar, and the number of differentially expressed modules, which is in pink. And you can see, for instance, for this highest concentration that you go from over 7,000 differentially expressed genes to 170 differentially expressed modules. Another nice thing to observe is that you still see a dose-response pattern in module activation. So here we see various dendrograms for the same compound, with on the left the lowest concentration and here on the right the highest concentration. And you actually see that the size of the dots, so which represent the modules, increases, but also the intensity of the color increases, indicating that with an increase in concentration, there is an increase in response. And because I project my data onto an already existing WGCNA map, I had to check whether there is a good correlation between the TG-Gates data, which is the basis for the WGCNA map, and my own data, which is the PHH experiment. So here you can see the Spearman correlation for two compounds, tunicamycin and diclofenac. And I used similar concentrations as those that were used in the TG-Gates study, but of course the primary human hepatocytes are different and also the sequencing platform is different. But taking that into account, we are very happy to see that there is a strong correlation, an R of 0.75, between these two data sets. And then also if we look, if we zoom in a bit on a specific module, so this is again methylpyrroline, and here I project the module scores for 3 modules, which are linked to oxidative stress. And then we see that in the TG-Gates data, these two go up. And as we see the same in my own experiment, and the other one goes down, and we see the same in my experiment. So we see a similar direction in module perturbation between the TG-Gates experiment and my new data. So this makes us more confident that we can actually use the existing WGCNA map to project data on. So then back to the full AOP. So here I've depicted the key events which could be coupled to modules, and the ones that are not are a bit more vague. So as you can see, not all these key events could be coupled to WGCNA modules, but we do not think that is of any problem, because we want to use this WGCNA as a screening method. And then if we progress in the testing battery, we can make sure to cover the other key events as well. And I do want to mention here that linked modules to key events do not per se form a direct measurement for the key event, but they are linked to it. So for instance, the sustained or regenerative proliferation. I'm not saying that there is a module that can be used to quantify proliferation, but the modules are enriched for AKT and ERK signaling, which are associated with proliferation. So yeah, it's more based on associations and not on direct measures. But after the verification, I ended up with 21 WTCNA modules that could be linked to this AOP. So I continued with these 21 modules and then made a correlation heat map. So for every chemical concentration combination, I looked up in literature whether there was evidence for oxidative stress induction in a cellular liver model. So this was mostly not primary human hepatocytes, it was mostly HepG2, but I've given those labels. So it's red if there's no evidence for oxidative stress, blue if I could not find any data for that specific combination of chemical and concentration, and then I used green if there was evidence for oxidative stress. Now if we look at the heat map, you see that based on the clustering, there are two major groups. And of course, these groups have subgroups within them, but there are two major groups. So this group here on the right mostly contains those chemical concentration combinations that, according to literature, induce oxidative stress in cellular liver models. And the one on the left mostly contains those combinations for which there is no evidence of oxidative stress in literature. And what is nice to see here, well, you can read it, but you have to trust me on my word for it, is that for all the chemicals for which we found at a certain dose level evidence for oxidative stress, there is indeed one concentration in this cluster, except for one. So one chemical, which according to literature should induce oxidative stress, we could not find any evidence for it in this transcriptomic screen. So the next step would be to generate the logistic regression model. I have to say that this is really an ongoing activity. So we really need to sort some things out here yet. But one of the things that you can use the logistic regression modeling for is to select your most predictive modules. So as a training data set, I here used again the TG Gates primary human hepatocyte data. And my input here is the data for these 21 modules within the TG Gates set. And then I performed stepwise model optimization, which basically means that the contribution of each module to the logistic regression model is scored. And whether removal of the module with the lowest contribution decreases the model AIC with at least 2. So the model AIC takes into account both goodness of fit and model complexity and is often used to compare different models. And this is an iterative process, and this is repeated until the model AIC is no longer decreased by 2 upon removal of the lowest contributing module. And then I ended up with 11 modules left. And you can here see which key events they are associated with. And these key events are spread throughout the AOP that I've shown before. So then ultimately, the idea is to apply this logistic regression modeling to predict the probabilities of a certain mode of action being activated by a chemical. So here you can see an example again from methylpyrroline, and you can see that it's mostly in these higher concentrations that the model is predicting a high probability of oxidative stress induction. And then if hypothetically we would do this for multiple modes of actions, you could compare these predicted probabilities and see, okay, which probability or which mode of action has the highest probability, and is this already of high probability at a lower dose or a lower concentration, then you can say, okay, maybe that's the main mode of action. And of course, you can also score the concentration response again in these predicted probabilities. But so far, we've only done this for oxidative stress, so the other line is purely. So what is next is to combine or integrate this screening method in a NAM-based testing strategy, in which we also take along the later key events to see if we can ultimately use that for hazard assessment of non-genotoxic carcinogens. Well, as I've already said with the previous slides, to compare probabilities for modes of actions, this would need to be repeated for more mode of actions. And furthermore, we want to further evaluate the regulatory application of this analysis method. And there are some ongoing efforts to link these modules to in vivo and integrate it into a quantitative AOP to really be able to have a quantitative assessment. And with that, I would like to thank all the people who worked with me on this project, and I would like to thank you for your attention.
Thank you, Kirsten. That was a really lovely presentation. I have a question for you, but we'll see. I'll ask it and we'll see if more questions come up. We have a few minutes, everyone, so please feel free to get your questions in. Kirsten, let me just explain where my question is coming from, because I think it needs a little background, which is that I know as we think about outlining AOPs that describe biology, and then taking that from an AOP to a testing strategy, we want to make sure, for practicality reasons, that we don't necessarily have to test every key event in a testing strategy. And so what I was thinking when I saw your diagram of the coupling of the modules to the key events, is that a way to understand whether certain key events are less maybe key than others? Is that information or that coupling an indicator that some of those key events that didn't couple might not be as important in an ultimate testing strategy, or is that not the case?
So I think you meant with the linking the modules to the key events?
Yeah.
Yeah, it's I wouldn't say that these key events are less important for the ultimate testing strategy. It's just that there was no transcriptomic signature that could be linked to it.
Okay.
Or at least not in the TG Gates data set. Okay. We enriched it.
Okay.
But I do agree that not every key event needs to be tested in a testing strategy.
Yeah. I think that's one of the challenges, is figuring out that question. There's a question coming from the audience. They want to ask if you could explain why it is called regenerative proliferation in your flowchart depicting oxidative stress leading to tumorigenesis.
Yes, so usually oxidative stress leads to damage, and then the body can regenerate to compensate for this damage. But if there's this balance between not being too much damage, but also not being too little damage, then this can, in the end, result in tumorigenesis.
Yeah, that is actually a really important mechanism of non-genotoxic carcinogenesis, right? I don't see any more questions, so I will go ahead and finish up here. Again, thank you, Kirsten, for your presentation. Thank you for sharing your work, and congratulations on your award. And as well to Damaris and Jerry, thank you so much for coming on to talk to us today about your work. Thank you all for listening and for your questions, and we will see you in May. Have a wonderful weekend.